B subgroups are very rare than A subgroup. The techniques done to differentiate weak B subgroups are agglutination with anti-ABH, presence or absence of ABO isoagglutinins in the serum, adsorption elution studies with anti-B, presence of B substance in the saliva, and molecular testing. This table will show first with the agglutination with anti-A, anti-B, anti-AB, and anti-H. So you will see that B phenotypes have B phenotypes strongly react with anti-B and anti-AB, with the 2+ reaction agglutination with anti-H. B3 phenotypes have mixed field agglutination with anti-B and anti-AB and has a 3+ agglutination reaction with anti-H. BX subgroup has a weak reaction with anti-B and anti-AB and has a 3+ agglutination reaction with anti-H. BM subgroup has 0 or weak agglutination with anti-B and anti-AB but has a 3+ reaction with anti-H. And BL has no reaction in all antibodies but has a 3+ reaction with anti-H. So in the serum, they all have anti-A antibodies in their serum. But unexpected na si B and B3 walang antibodies, si BX may weak anti-B antibodies, si BM none sa anti-B, while si BL sometimes may weak and anti-B antibody siya. So in the saliva, may presence of BH sa B, B3, and BM, and only H sa BX and BL. So, in the presence of B transferase in serum, walang B transferase sa BX and BL. Pero may weak, meron sa B3 and BM, pero weak positive only. So again, B subgroups should be identified correctly because, for example, like BL, ang reaction niya is parang O blood group. So, pag ito siya mag-donate doon sa O blood group, magkakaroon talaga ng hemolysis, leading to maybe death of the patient. Bombay or OH phenotype. Bombay phenotype was first reported by Bende in 1952 in Bombay, India. The individuals will inherit the lowercase h gene and thus having the lowercase hh genotype. ABO are not expressed because it lacks the H antigen. So they are also called OH or H null. Bombay phenotype has the lowercase hh genotype. No H antigens are formed, therefore no A or B antigens are formed. Phenotypes as blood group O, anti-A, anti-B, and anti-AB, and anti-H are present in the serum. So therefore, they can only be transfused with blood from another Bombay. The characteristics of Bombay phenotypes: absence of H, A, and B antigen, so no agglutination with anti-A and anti-B or anti-H lectin. Presence of anti-A, anti-B, anti-AB, and a potent wide thermal range of anti-H in the serum. ABH non-secretor. No A, B, or H substances present in the saliva. So, absence of alpha-2-L-fucosyltransferase, the H enzyme, in serum, and H antigen on the red blood cells. Presence of A or B enzymes in serum, depending on ABO genotype. A recessive mode of inheritance, identical phenotypes in children but not in parents. RBCs of the Bombay phenotype will not react with the anti-H lectin. RBCs of the Bombay phenotype are compatible only with the serum from another Bombay individual. Para-Bombay phenotypes are rare phenotypes which the RBCs lack or have small amounts of H antigen. This is because the FUT1H gene is silenced or mutated. The production of alpha-2-L-fucosyltransferase will be reduced, thus making small amounts of ABH antigens. This phenotype is only detected using adsorption elution techniques only. Before we move on to the next topic, let me show you the compatibility of blood types. So here we have the recipient and here we have the donor. So AB positive can receive blood from all blood types. AB negative can receive blood from O neg, B neg, A neg, and AB negative individuals. A positive individuals can receive blood from O neg, O positive, A neg, and A positive individuals. A negative individuals can receive from O negative and A negative individuals. B positive individuals can receive blood from O negative, O positive, B negative, and B positive individuals. B negative recipients can receive blood from O negative and B negative donors. O positive recipient can receive blood from O negative and O positive donor. And lastly, O negative can only receive blood from O negative individuals. So, so you will see here that the AB positive individuals are the universal recipient because they can get blood from any blood type. And O negative is the universal donor because O negative individuals can donate blood to all blood types. But take note that O negative recipients only receives O negative blood only, nothing else. Now let us proceed to the next topic. ABO discrepancies. Technical errors can cause ABO discrepancies, like mislabeling, failure to add reagents, addition of incorrect reagents or sample, or reagent contamination. So the resolution for uh, mislabeled tubes is repeat collection. If there is a failure to add reagents or addition of incorrect reagents or sample, you will um, repeat the procedure. If the reagent is contaminated, you will change the reagent with another lot number. Here are more examples of technical errors. So, incorrect or inadequate identification of blood specimens, test tubes, or slides. Cell suspension either too heavy or too light. Clerical errors or incorrect recording of results. A mix-up in samples. Missed observation of hemolysis. Failure to add reagents. Failure to add sample. Failure to follow manufacturer's instructions. Uncalibrated centrifuge. Over-centrifugation or under-centrifugation, contaminated reagents, and warming during centrifugation. ABO discrepancies have four categories. First, the group 1 discrepancies, where there are unexpected reactions in the reverse grouping due to weakly reacting or missing antibodies. You will encounter this with newborns because ABO antibodies are not yet detectable until 3 to 6 months. With elderlies because elderlies have depressed ABO antibodies. You will also see this with patients that have leukemia and lymphoma because they demonstrate hypogammaglobulinemia, which prevents the immune system to make enough antibodies. You'll also see this in patients with agammaglobulinemia and immunodeficiency diseases. Because these are inherited disorder with low level of immunoglobulins. You will also see these discrepancies in patients with bone marrow and hematopoietic progenitor stem cell transplants. In plasma transfusions, where antibodies are diluted, and in individuals with ABO subgroups. So the resolution for these discrepancies, first, you will incubate the serum with A1 cells and B cells at room temperature for 15 to 30 minutes. And then, if after centrifugation there is still no agglutination, the mixture can be incubated at 4 degrees Celsius for 15 to 30 minutes, and then there should be a test for autocontrol and O cell control. Group 2 discrepancies. There will be an unexpected reaction in the forward grouping due to weakly reacting or missing antigens. You will see this in individuals with A and B subgroups, patients with leukemias and Hodgkin's disease, where antigens are weakened or depressed, and patients with acquired B phenomenon. So, patients with acquired B phenomenon, they acquire a B-like antigen which weakly reacts with anti-B anti-sera. So, this is common in colon cancer. The resolution is incubate red cells with anti-sera at room temperature for 15 to 30 minutes. And if there are still no reaction after centrifugation, the mixture can be incubated at 4 degrees Celsius for 15 to 30 minutes. Test for autocontrol and O cell control. And RBCs can be pre-tested with enzymes and retested with anti-sera. Table 619 is an example of ABO discrepancy caused by an acquired B antigen. So, you will see here that in the forward grouping, may 2 plus reaction si patient with anti-B. But sa reverse grouping, meron siyang 4 plus reaction sa B cells and 0 reaction sa A1 cells. So, based sa reverse grouping, A dapat ang blood type niya. So, bakit may 2 plus dito sa anti-B? So, this is because the patient acquired B antigen. So, patient's RBCs have acquired a B-like antigen that reacts with reagent, anti-B, and is associated with cancer of the colon or other diseases of the digestive tract. So, the resolution for this discrepancy, number 1, acidify anti-B reagent to a pH of 6. Second, run DAT. And third, run autocontrol. In table 620, this is an example of ABO discrepancy caused by low prevalence antibodies in the reagent anti-sera. So, napapansin nyo, may 1 plus reaction sa anti-B. Then, sa reverse naman, 4 plus reaction in both A1 and B cells. So, the reaction here is with anti-B in forward type is due to agglutination between a low prevalence antibody in reagent, anti-B, and the corresponding antigen on the patient cells. So, meaning may problem sa reagent or sa anti-sera. So, ang resolution nito is use a different lot number for reagent anti-B. There is also we call blood group specific soluble substances. They neutralize reagents anti-A and anti-B, leaving no antibody to react with the patient's red cells, resulting to false negative or weak reaction in forward grouping. So, ang solution nito is you should wash patient's cells with saline. Group 3, discrepancies. These are discrepancies between forward and reverse grouping caused by protein or plasma abnormalities resulting to rouleau formation or pseudoagglutination. You will see these discrepancies in elevated levels of globulin, like, for example, multiple myeloma and Waldenstrom macroglobulinemia, where cancer cells make large amounts of abnormal proteins. You will also see this in elevated levels of fibrinogen with plasma expanders. Plasma expanders are colloidal solutions that expand circulating blood volume. And lastly, you will also see this in Wharton's jelly. Wharton's jelly is a gelatinous substance within the umbilical cord that contains fibroblasts and macrophages. For the resolution, first, check the slide microscopically. So you will check if there are rulu formation. So if there are rulu formation, wash red cells with saline 3 times. But in Wharton's jelly case, you will wash the red cells 6 to 8 times. But in cord blood, you will still use forward grouping. You will not use the reverse grouping because antibodies might be from the maternal origin. And lastly, run antibody screen. And the last category is the group 4 discrepancies. Discrepancies between forward and reverse grouping caused by miscellaneous problems. So these miscellaneous problems are cold reactive autoantibodies, circulating of RBCs more than one ABO group, unexpected ABO isoagglutinins, and unexpected non-ABO alloantibodies. Table 622 is an example of ABO discrepancy caused by cold autoantibodies. So the patient's blood group supposedly is B. So kasi dito sa anti-B is 4 plus. Then sa A1 cells, reverse grouping 4 plus din. Pero bakit may 2 plus reaction sa anti-A and 2 plus reaction sa B cells? So reaction with anti-A in forward type is due to spontaneous agglutination of antibody-coated cells. Then, sa reaction sa B cells in the reverse type is due to cold autoantibody. So, ang resolution for this is wash patient cells with warm saline and retest. Second, run DAT and autocontrol. And third, run antibody screen. Sa Table 623 is an example of ABO discrepancy caused by an unexpected ABO isoagglutinin. So in this table, blood type supposedly ng patient is A2B. So ang reaction niya sa forward grouping is 4 plus sa A and 4 plus din sa anti-B. Sa reverse naman, negative sa B cells but may 1 plus sa A cells. So take note, A to B individuals have anti-A1 sa kanilang plasma. So reactions with patient serum are due to anti-A agglutinating A1 reagent, red blood cells. So, ang resolution for this is test cells with anti-A1 lectin. Second, test serum with A1, A2, and O cells, and third, run an autocontrol. Dito sa last table, 624, it is an example for ABO discrepancy caused by an unexpected non-ABO alloantibody. So here, ang patient's blood group supposedly is AB. So sa forward grouping, 4 plus and 4 plus ang result niya. But sa reverse grouping, may 1 plus reaction in both A1 cells and B cells. That supposedly walang reaction. So this is reactions with patient serum are due to non-ABO alloantibody agglutinating an antigen other than A1 and B on reagent red cells. So ang resolution po nito is run an antibody screen and panel. Let's move to our next slide. So this figure is a simplified summary of ABO discrepancies. So causes of ABO discrepancies sa forward typing are mixing or weak antigens. So pwede siya sa A or B subgroups or sa mga leukemia. Extra antigens. So makikita siya sa acquired B. For example, yung may mga colon cancer, Lulu formation, cold autoantibodies, and Wharton's jelly. Mixed field reaction sa group O transfusions, stem cell transplants, and A3 or B3 phenotypes. Sa reverse grouping naman, may dito sa mixing or weak antibodies, so makikita siya sa newborns, elderly, and immunocompromised na individuals. Dito sa may mga extra antibodies, so ito sa anti-A1, may cold alloantibodies, Lulu formation, cold autoantibodies, and passively acquired antibody. Example, plasma exchange or mismatch na platelets. And that's all for our lesson for this week. And here, I will share to you some videos for the ABO grouping methods. The first one is the video for forward and reverse grouping tube method. And the next link is for the forward grouping slide method. And the last link is for the forward and reverse grouping method, gel card method. So, in this video, for the gel card method, hindi na nag-wash ng red cells yung medtech. So, I just want to remind you that kailangan talaga i-wash ang red cells para maiwasan yung ABO discrepancies. This is the reference for our lesson this week.